Vin Future Prize, a new global science and technology prize for humanity from Vietnam. One Vin Future Grand Prize of $3 million. 3 additional special Vin Future prizes valued at $500,000 each. Vin Future Prize honors science and technology work that creates or has a high potential to create meaningful change in the everyday lives of millions of people. Join us to make a change for a better future.
Ladies and gentlemen, this is the event in the framework of Vin Future SciTech Week, Vin Future Prize 2023. Ladies and gentlemen, Vin Future Prize is a prestigious global science and technology award with a commitment to science and technology at the service of people. With the central theme, Boundless Unity, the Vin Future Prize 2023 wishes to find scientific solutions to help solve urgent global problems and at the same time inspired to spread widely the good values that science and technology can bring with many new fields to many new countries and territories, especially vulnerable areas, in which it addresses 4 major key issues: health and food, environment, sustainable energy, as well as the application of technology in all aspects of life. Therefore, biomedical discussion session with the theme, Advancing Precision Immunology Therapy for the Treatment of Autoimmune Diseases, within the framework of activities towards the Vin Future Prize 2023, is a forum for the world's leading scientists to discuss the big problems of global health and the potential for development in medicine for human service. And to come to this event today, we are honored to have our distinguished guest. We want to extend a warm welcome to the event Associate Professor PhD Nguyễn Thị Liên Hương, Deputy Minister of Health. Let us welcome Dr. Nguyễn Ngô Quang, Deputy Director, Department of Science, Technology and Training, Minister of Health. Welcome, sir. And last but not least, let us welcome Professor Chí Văn Đặng, the Scientific Director of the Ludwig Institute for Cancer Research, member of the Vin Future Prize Council, and the chair of today's discussion. And to start this afternoon event, we would love to invite to the stage Deputy Minister of Health, Associate Professor PhD Nguyễn Thị Liên Hương, to be on the stage for her speech. Let us welcome our distinguished guest. The stage is yours.
Thưa bà Lê Thúy Anh, Tổng Giám đốc Hệ thống Y tế Vinmex, thưa toàn thể quý vị đại biểu, khách quý cùng toàn thể các nhà khoa học trong nước và quốc tế. Trước hết, thay mặt lãnh đạo Bộ Y tế, tôi xin nhiệt liệt chào mừng các quý vị đại biểu, khách quý, các nhà khoa học đến tham dự chương trình tọa đàm Khoa học vì cuộc sống, phiên thảo luận về thúc đẩy miễn dịch học chính xác để điều trị các bệnh rối loạn tự miễn. Tôi xin gửi tới toàn thể quý vị đại biểu, khách quý lời chào trân trọng, lời chúc tốt đẹp nhất. Thưa toàn thể quý vị đại biểu, khách quý, chúng ta đang có mặt tại tọa đàm trong khuôn khổ chương trình lễ trao giải Vin Future, một trong những giải thưởng về khoa học danh giá nhằm tôn vinh các phát minh khoa học có tác động đến chất lượng cuộc sống toàn cầu. Hôm nay là một dịp vô cùng ý nghĩa khi các nhà khoa học trong nước và quốc tế quy tụ tại đây, cùng nhau thảo luận về chủ đề đang rất được quan tâm, thúc đẩy miễn dịch học chính xác để điều trị các bệnh rối loạn tự miễn. Bệnh rối loạn tự miễn được công nhận lần đầu vào năm 1900. Trải qua hơn một thế kỷ, con người đã có những bước tiến đáng kể trong hiểu biết và quản lý tình trạng bệnh. Tuy nhiên, việc làm sáng tỏ nguyên nhân gây bệnh và tiến triển phức tạp của các bệnh rối loạn tự miễn vẫn luôn là thách thức lớn, tiêu tốn nhiều thời gian, công sức của các nhà khoa học. Nhân loại biết đến khoảng 100 loại bệnh tự miễn trong tổng số hàng nghìn bệnh lý hiếm gặp. Đáng chú ý hơn, kể từ sau đại dịch Covid-19, các nghiên cứu cho thấy nguy cơ mắc các bệnh lý tự miễn tăng lên từ 20 đến 50% sau khi nhiễm virus corona. Tại Việt Nam cũng ghi nhận tỷ lệ người mắc các rối loạn miễn dịch sau COVID-19 gia tăng. Nguồn dữ liệu về phương pháp điều trị còn hạn chế, trong khi tỷ lệ mắc các bệnh tự miễn ngày một tăng cao. Rối loạn tự miễn cho đến nay đã và đang gây ảnh hưởng trực tiếp cho hàng trăm triệu người trên thế giới về mặt sức khỏe, kinh tế và xã hội. Thưa toàn thể quý vị đại biểu khách quý, qua 2 mùa, giải thưởng VinFuture vinh danh những công trình nghiên cứu xuất sắc, góp phần giải quyết các thách thức của nhân loại với sức lan tỏa mạnh mẽ. Những chủ đề nghiên cứu, các hội thảo còn mang tầm ảnh hưởng to lớn, tạo sức hút và truyền cảm hứng đến cộng đồng các nhà khoa học trong nước và toàn cầu. Đứng trước thực trạng còn nhiều hạn chế trong việc điều trị các bệnh rối loạn tự miễn, đặc biệt tại Việt Nam, với nhiều khó khăn về các loại thuốc điều trị, tọa đàm này là điều kiện để kết nối các nhà khoa học, hợp tác tạo nên những bước tiến mới mang đến niềm hy vọng to lớn cho những bệnh nhân bị rối loạn tự miễn trên toàn thế giới nói chung và tại Việt Nam nói riêng. Thay mặt lãnh đạo Bộ Y tế, tôi ghi nhận những nỗ lực của Quỹ VinFuture trong việc tổ chức diễn đàn khoa học với mục tiêu và sứ mệnh đầy ý nghĩa này. Chúc cho tọa đàm và giải thưởng VinFuture năm nay thành công rực rỡ. Xin trân trọng cảm ơn.
Thank you so much, Deputy Minister of Health, Associate Professor PhD Nguyen Hương, for your speech. Thank you so much. Ladies and gentlemen, and now I would love to focus your attention to the stage on the screen for the short video for the opening video today. Video, please.
According to the Human Disease Database, there are approximately 40,000 diseases known to humans, a quarter of which are rare diseases, with 90% still lack effective treatment methods. Among them are autoimmune diseases, a group of over 100 incurable rare diseases with unclear underlying mechanism. Autoimmune diseases occur when the immune system's normal function is disrupted, leading it to mistakenly attack the body's healthy cells. To solve this challenge, scientists have been developing a new therapy, precision immunology therapy. This is an innovative method with the potential to enhance and reprogram our immune systems to target the specific disease-causing cells and molecules. Today, various next-generation treatment techniques are being developed, including cell-based therapies, antigen-specific therapies, novel protein approaches, and even new cytokine therapeutics, all of which will be explored further in this symposium. Presenting and chairing this panel, Professor Chi Van Dang, the Scientific Director of the Ludwig Institute for Cancer Research and Bloomberg Distinguished Professor at Johns Hopkins University. He is also a member of the VinFuture Prize Council, a member of Biden Cancer Moonshot Blue Ribbon Panel. Professor Zhang Su Chun, who has dedicated over 20 years to studying the pathogenesis of degenerative arthritis. His findings have been published in several world's leading journals. Professor Shimon Sakaguchi, the Robert Koch Prize winner. He has discovered the population of regulatory T cells that caused a variety of immunological disorders, including autoimmune diseases. Professor Pascale Cossart, Perpetual Secretary of the Academy of Sciences, France, and member of the VinFuture Prize Council. She has received many prestigious awards, including the Robert Koch Prize or the Selman A. Waxman Award in Microbiology. Assistant Professor Nguyen Van Dinh, Head of General Medicine Department at Vinmec International Hospital, with over a decade of expertise in allergy and clinical immunology. Under the theme Boundless Unity, VinFuture Prize Award Ceremony 2023 aims to discover outstanding science and technology achievements. This symposium will mainly discuss advancing precision immunology therapy to treat autoimmune disorders, featuring world's leading scientists. Let's come together to unlock a future where healthier lives know boundaries.
that, we have a brief introduction to the exciting field of precision immunology therapy, which holds the potential to transform the way we treat autoimmune diseases. And that's why we are here today to delve into this inspiring topic. And the biomedicine session today includes 3 main parts: presentation, panel discussion, and Q&A, focusing on the applications of precision immunology therapy, the treatment of autoimmune diseases, as well as the challenges and opportunities for the technology and its application in Vietnam. And to commence today's discussion, we would love to invite to the stage Professor Chi Van Dang, the Scientific Director of the Ludwig Institute for Cancer Research, the member of the Vin Future Prize Council, to be on the stage and give us some insight into the introduction of the precision immunology. Professor, please. And now it's your time. Thank you.
Noon. I'm going to give you a general introduction to the subject so that we're all on the same page before the other distinguished speakers get to their talk. So what I'm going to tell you in the next few minutes is the distinction between self and non-self. And so if you look at the cartoon that's shown on the left, what I illustrated here is essentially what you would find in the gut epithelium, in your gut, in your colon. You'll see there's cells that separates the outside world, the non-self, where all the bacteria and other viruses as well as fungus would be residing. And this is a topic that Professor Kozart would be expounding on later. Now you can see the barrier of cells in the pink that holds the outside world from the inside world, being self. If that barrier is breached and some of the organisms actually make it into the tissues, what happens is that you'll see their cells, innate cells that will react to these infiltrating organisms, such as neutrophils and macrophages, that will react to these and try to destroy this organism. Now, if there's still a breach, we have a second mechanism, which is the adaptive immune system that consists of T cells and B cells. And what I show in the middle is one of the effector cells that can get rid of infected cells or cells that are abnormal. And the way these cells work, the T cells, is they have receptors, these proteins on the surface, that can recognize what we call MHC molecule on the target cell that presents a protein fragment on its surface. So each cell in our body can process various proteins and present it to the surface. Our immune system then survey and see if they're foreign, that is, non-self versus self. Now, how do you distinguish between self and non-self? If you look on the far right, the thymus gland, which sits in the upper part of our chest, when we were younger, is actually a site where these T cells are educated. So what you'll see at the top of the figure are specialized cells in the cortex of this organ, where the T cells comes in and gets activated, and then they travel into another part of this organ called the medulla. In here, we have very specialized cells. They're called epithelial cells of the thyroid gland, especially the medullary one. And these cells are very special because they produce peptides that are found in our organs. So basically, any T cells that recognize a peptide from a normal organ, they will be eliminated by cell death so that you don't react to self, or they become Tregs. These are CD4s, which you'll hear from Professor Sakaguchi. Now, once the T cell's been educated, what do? If the T cells then gets further stimulated by cells we call dendritic cells that recognize, for example, COVID virus, and presents the COVID protein to the T cells then begin to reactivate it, proliferate, and then kill the infected cell so that you eliminate the infection. However, the problem is that these cells can react to cells in our body that might have proteins that mimic, say, a virus. And that, at the bottom, you'll see that our T cells begin to destroy our own cells, and that causes autoimmune cell death. Now, the B cells is another type of lymphocytes shown in the middle, is the type of cells that are educated differently. They give you a second type of defense, which is to produce proteins that can bind to viruses and eliminate them. Now, if you see these cells, they're being educated by T cells, and they can then differentiate into plasma cells, the ovoid cells. They produce these proteins and bind to viruses and neutralize them. The problem is that some of these B cells actually can make antibodies against proteins that are made by your normal organs. And I illustrate a disease called pemphigus vulgaris, where the B cells actually can make antibodies that attack the skin and causes this horrible blebbing that is very painful. But technology actually has really moved very rapidly forward in how to get rid of the B cells to eliminate them from causing autoimmune diseases. And based on chimeric antigen receptor T cells that target B cells themselves to treat leukemias or lymphomas, that these now have been used in clinical studies showing that they can be effective against a number of different autoimmune diseases. A new development, which is using the same concept to make a chimera of a T cell receptor that is fused to the antigen, and the antigen then, the red dot at the bottom there, can be recognized by the B cells. The T cells are professional killers. They then kill off all the B cells that makes the antibodies, thereby eliminating the autoimmune disease. Now, lastly, I just want to, because I'm actually a cancer researcher and oncologist, that there's been advances in immunotherapy for cancer. So how does that work? Well, T cells can also recognize cancer cells for the same reasons as cancer cells make abnormal proteins. But when T cells are activated, they can be turned off, what we call checkpoints, shown at the bottom, such as CTLA-4 or PD-L1, PD-1 axis. So drugs have now been made to block these negative feedback and activate the T cells to kill cancer cells. And this has been very effective for melanoma, a skin cancer. The problem is, you look in the middle and the far right, is I illustrate here a patient with metastatic melanoma that got treatment with these two antibodies. On the left are all the disease that's found in the body by PET scan. On the right is when the patient's been treated with these antibodies to allow for our immune system to go after the cancer cells. The problem is, those immune cells can also attack our melanocytes, and that causes not a very dreadful autoimmune disease, but you can see there where you get blanching where the melanocytes are killed off. So clearly, our immune system is a double-edged sword. On the one hand, it protects us from infiltrators. On the other hand, self-reaction causes autoimmune disease. So I just want to highlight that you'll now hear from the other speakers that will talk about. Dr. Chung will talk about inflammatory in autoimmune diseases. So thank you, and I would like to ask for Dr. Chung to come up to the stage.
Thank you so much, Professor Chi Van Dang, for your insightful presentation. Thank you so much. Ladies and gentlemen, and now I would love to invite to the stage Professor Jang Soo-chun from Gwangju Institute of Science and Technology, who will offer us a comprehensive view of targeted therapies in the treatment of autoimmune disorders. And for your information, Professor Jang Soo-chun has studied the pathogenesis of degenerative arthritis for more than 20 years and published his results in many of the world top journals. And now, the stage is yours. Welcome, Professor Jang Soo-chun.
Thank you. Thank you for having me at this symposium. My name is Jang Soo-chun, actually. My research area is the molecular mechanisms of osteoarthritis. So based on our previous discoveries that certain molecules are common therapeutic targets for both osteoarthritis and rheumatoid arthritis, I'm going to pose a couple of questions regarding targeted therapy and the precision medicine in rheumatoid arthritis. So the osteoarthritis is a typical degenerative joint disease and characterized by the destruction of cartilage tissue in joints. But the disease is not simply a wear and tear disease, but it can be actively induced by numerous risk factors. For instance, low-grade chronic inflammation is implicated in aging-associated osteoarthritis. And metabolic osteoarthritis can result from mechanical stress in joint tissues, and the metabolic osteoarthritis, of course, linked to local or systemic metabolic disorders. So the osteoarthritis is actually a whole joint disease involving not only cartilage destruction, but also many other symptoms such as inflammation, osteophyte formation, and subchondral bone sclerosis. However, we do not have any effective disease-modifying drugs for osteoarthritis until today. Rheumatoid arthritis is a chronic inflammatory autoimmune disease, which also leads to joint damage. And the disease is highly heterogeneous. So although most of RA patients are seropositive, I mean, they have antibodies in the blood, but still a significant portion of RA patients are seronegative. And up to 40% of RA patients exhibit little to no response to current medication. So the pathogenesis of rheumatoid arthritis is very complex. It involves various cell types and cytokines with multiple steps, such as production of autoantigen and activation of adaptive immune response, production of autoantibody, synovitis, and finally, joint damages. So I thought precision medicine and targeted therapy might be a very useful approach to treating the rheumatoid arthritis patients. Both osteoarthritis and rheumatoid arthritis develops from completely different etiologies. So although they share common symptoms such as cartilage destruction and inflammation, they also manifest distinct or even opposite symptoms. So as I mentioned earlier, we were able to identify several molecules which are common targets for osteoarthritis and rheumatoid arthritis. The example include HIF2-alpha, hypoxia-inducible factor 2-alpha, which is a transcription factor, and GB8, which is a zinc import on cell membrane. Oh, I'm sorry. Oh. In osteoarthritis, we were able to demonstrate that HIF2-alpha is upregulated in osteoarthritic chondrocytes. And the upregulated HIF2-alpha acts as actually a master transcription factor in osteoarthritis. That's what we believe. And HIF2-alpha induces expression of many catabolic effector molecules such as cytokines, chemokines, and adipokines. HIF2-alpha also induces expression of catabolic mediators such as the enzymes which regulate cholesterol metabolism and mRNA metabolism. And other transcription factors are also targets for HIF2-alpha in chondrocytes. So we demonstrated HIF2-alpha specific functions in chondrocyte by using through gain of function and loss of function studies in a mouse model. In addition to regulating osteoarthritis, we found that HIF2-alpha in synovial cells regulate the development of experimental rheumatoid arthritis. So in HIF2-alpha in synoviocyte, I mean FLS, it regulates the differentiation of Th17 cells, destruction of cartilage and bone, and the synovial hyperplasia by regulating the specific target molecules, which include rancor, TNF-alpha, or IL-6. However, we do not know at these times the expression profiles of HIF2-alpha and the cell type specific functions of HIF2-alpha in various immune cells. We also found that the zinc, GB8, MTF1 axis in chondrocyte is a critical pathway in osteoarthritis. So we found that HIF2α is regulated in osteoarthritic chondrocyte, and it causes influx of zinc ion into chondrocyte and thereby activate transcription factor called MTF1, which is a zinc ion dependent transcription factor. And the activated MTF1 then induces the production of matrix degrading enzymes such as MMP3 and MMP13, which is a critical enzymes in the destruction of cartilage tissues. So this function is also chondrocyte specific, as we demonstrated by gain of function and loss of function approaches in a mouse system. And finally, we discovered that zinc influx, the CD4 T cell in CD4 T cells, which GBA-mediated zinc influx in CD4 T cells is critical for the development of experimental rheumatoid arthritis. So we were able to identify that many cell types in the synovial cells of express the increased protein levels of GBA, and actually nearly all CD4 T cells exhibit increased protein levels of GBA. And we also demonstrated CD4 T cell specific GBA knockout mice demonstrated effective inhibition of the development of experimental rheumatoid arthritis. We also found that GBA actually governs the effective stage of CD4 T cell activation through TCR signaling. So as I mentioned earlier, I would like to raise two questions regarding the rheumatoid arthritis. So the first question is the possibility that if we can use HIF2α or GBA in biomarkers for precision medicine, more specifically the stratification, diagnostics, and the prognosis, and therapeutic precision medicine. I thought it is possible, but to effectively utilize them as biomarkers in rheumatoid arthritis, we need a more comprehensive characterization, such as single-cell transcriptomics and proteomics. The other question is the potential use of these molecules, these target molecules, in targeted therapy. As I mentioned, not all RA patients responded to current medications. So the current medication include conventional synthetic DMARD, disease-modifying anti-rheumatic drugs, and the biologic DMARD, which is typically monoclonal antibodies or recombinant proteins against the cytokines and immune cells. And the last one is targeted synthetic DMARD, which is developed against the tyrosine kinase. So the development of targeted synthetic drugs for HIF2α and GBA might be very promising for the treatment of both rheumatoid arthritis and osteoarthritis. But I should mention at this time that this remains as a hope rather than a reality. Thank you very much for your attention.
We sincerely thank you, Professor Jang Soo-chun, for delivering a highly enlightening presentation on the current application and upcoming trends in targeted therapies. And ladies and gentlemen, and now let us welcome to the stage Professor Shimon Sakaguchi from Osaka University, whose lecture will delve into the pivotal role of regulatory T cells in the treatment of immunological diseases. Professor, please. Thank you.
Thank you. Well, so already Professor Jiban Dan introduced to us our immune system, which is not supposed to react with ourselves if it aggresses our the cellular or tissue components, and we have autoimmune diseases. So it is estimated that at least 5% of the population worldwide is afflicted with some sort of autoimmune diseases, including in various organs. joint or pancreas and type 1 diabetes or other almost all tissues and organs, and potentially we have autoimmune diseases. So how we can understand the mechanism of autoimmune diseases? That has been the challenge for modern immunology. So historically, one mechanism which was proposed in the 1960s, that was the lymphocytes, the upper one, when they develop, and then they react with self-antigen, and then they'll die by apoptosis. So it is called deletion. And second mechanism is the inactivation. So lymphocytes recognize the self-antigen, but they become inactivated. And so it is called anergy in immunological term. And third one, that is suppression. So our immune system, the potentially dangerous lymphocytes, they are present in normal individuals, but their activation or expansion is controlled by another subset of T cells. Now we call them regulatory T cells. So what has been challenging to immunologists, that was how we can induce these autoimmune diseases by manipulating the left side mechanisms. So what we found is that the third mechanism, suppression, when we remove the regulatory T cells, then we can induce the animals that spontaneously develop these various autoimmune diseases. So this is right side is what we did. So here are normal mice, and then they are healthy. So if you take out the spleen is lots of lymphocytes, including T cells and B cells, and we remove the regulatory T cells defined by CD25. This is the marker on the cell surface, and FOXP3. This is the marker in the nucleus. And in human and animals, and even now, we can define regulatory T cells by FOXP3, which is a transcription factor, master controller of the Treg development and function. And CD25, this is an IL-2 receptor. So here, the 10% of the population in the spleen and 5% of the population in the thymus, they are regulatory T cells. So if you remove them and then transfer into a T cell deficient mice, all T cells gone, and if you transfer a Treg deficient lymphocyte, then you can make the animals which are deficient in regulatory T cells or devoid of regulatory T cells. And these mice spontaneously develop various autoimmune diseases, thyroid, stomach, or pancreas in type 1 diabetes. So then based on these findings by us and others, now our understanding is that normal thymus here, they are producing T cells. So highly selective self-reactive one, very dangerous one is depleted. They'll die by apoptosis. But the majority of hazardous self-reactive T cells, they are produced by the thymus. But the thymus also produces the regulatory T cells, which are specialized for immune suppression. So if you remove regulatory T cells, you will have autoimmune disease, allergy, or IBD, inflammatory bowel disease, which is an immune hyperreaction to commensal bacteria in the intestine. And also, by reducing them, and then you can have an enhanced immune responses. So it's good for the tumor immunity, cancer immunity. But at the same time, if you remove them, autoimmunity happens. And so both happens. And also chronic infection, that's good. And then also enhancing or expanding them, you can strongly suppress immune responses. So the transplantation from organ transplantation from others, they can be accepted. And fetal-maternal tolerance. Why mothers they have the non-self fetus, but they don't reject them? It is called fetal-maternal tolerance. even now, we don't know the exact mechanism of fetal-maternal tolerance. But if you look at the Treg at the pregnant, say, placenta, there are lots of regulatory T cells and suppress the abortion. Regulatory T cells, majority of them produced by the thymus, we call the Treg, thymus-derived. And also some regulatory T cells can be produced in the intestine, for example. So we call them P-Treg, peripherally derived Treg, and also I-Treg. It is induced in vitro. So the question for treatment of autoimmune disease, that is how we can achieve antigen-specific suppression. For example, steroid or tacrolimus or cyclosporin A, they are very good immunosuppressive drug, but then suppress all the immune responses. So if you use for treatment of autoimmune disease, at the same time, the patient become sensitive or vulnerable to infectious diseases. So how we can achieve antigen-specific, just suppress the autoimmune responses, but not the antiviral immune responses. That is what we are working on, we and then others are working on. So to do that, the one way is natural Treg. They are present in our body. So how we can expand them, and then perhaps we can make a Treg dominant state for immune suppression or tolerance. So low-dose IL-2, that is under clinical trials in the United States and also Europe. So Treg expressing the high affinity IL-2 receptor. So they are very sensitive to IL-2. IL-2 is a very important cytokine. So just low dose. Low dose means the Treg can expand, but other T cells, they don't. So we can make a Treg dominant state. The second approach is they are present in our body, so take them out. And the ex vivo, the outside of the body, you can expand them with antigen stimulation in the presence of IL-2, and then transfer back to the patient. So that is adoptive cell therapy. And also, just recently, a CAR T technology has been developed. So it's good for anti-cancer therapy. So the idea is, if the Treg, then they are manipulated to go to the particular inflammation or in the recognized particular antigen. So that is called CAR Treg the technology. So that is under clinical trial now. Third approach, which we are working on, the ideal way of immune suppression is, so the lymphocyte, which are mediating disease, then if we can convert effector T cells, bad guys, into Treg, that kind of technology, if it is developed, it's an ideal way of antigen-specific suppression. So we are developing that kind of drugs which can effector T cells, disease-mediating cells can be converted to P-Treg, peripherally derived Treg, in the body. And then another approach is just take the effector T cells out from the blood and then convert them in vitro. For example, FOXP3, this is an important transcription factor, induce it, and also epigenome to make the Treg function stable. So these together, now you can make an induced Treg in vitro and then expand them and then transfer back to the patient. Now, then you can induce antigen-specific suppression. So various approaches are now taken in the world. And then in the future, what we hope is that not only a cell therapy, but taking a drug when you are diagnosed to have an autoimmune disease, and take drug, and then some Treg can expand or converted from effector T cells into Treg, and then induce the long-term tolerance or cure autoimmune disease. Thank you.
Thank you so much. Thank you, Professor Shimon Sakaguchi, for having shed light on the role of T cells in the treatment of immunological diseases. And now we're coming to the fourth presentation today, which is presented by Professor Pascale Cossart. She's from Pasteur Institute as Perpetual Secretary of the Academy of Science in France, and she is a member of the VIN Future Prize Council. Let us welcome and give a round of applause for the Professor Pascale Cossart.
Okay, I'm very happy to be here and to introduce a new component in this search for treating autoimmune disease. And this last component is the microbiota, the microbiome. So, sorry.
We need next.
Okay. So what are microbiomes? Microbiomes are assemblies of microbes, bacteria, viruses. They reside in various parts of the body, including the gut and the skin. And our bodies harbors 10 times more microbes than its number of cells. 100 trillions, 10 to the 14th, maybe 10 to the 13th bacteria are in our body, among which 10 to the 13th are in the intestine. So what is important is to realize that, in fact, the composition of the microbiomes is different in the hair, in the skin, in the vagina, in the oral cavity, in the stomach, in the intestine, in the colon. So what are the classical functions of the gut microbiome? I think you all know that microbiota participate to the last step of the digestion of food products. They produce metabolites and vitamins. They participate to the development of the immune system. They protect against pathogens by producing mucus or production of antimicrobial peptides. And in addition, it's well established now that the microbiota in the gut communicate with the brain, affecting mood and behavior. So I think it's important to realize that microbiota are very important. And what is important is both the richness and the diversity. And we know that the composition of the microbiota varies in different parts of the gastrointestinal tract, and also that it varies with the geographic origin of the human which are analyzed. For example, if you see on the slide, for example, the Italian people have more bacteroidetes than firmicutes. I'm going to try to show you that. I don't think okay, I don't think, okay. So the Italian people have more bacteroidetes than firmicutes. In contrast, in Africans, you have more bacteroidetes than firmicutes. And if you analyze these people from Venezuela, you will see that the diversity in this population is amazing. So the microbiome composition is really, really affected by the alimentation, by the nutrition. So as you can see here, if you, I'm sorry. If you can see on the left, if you have an alimentation, if you have a diet which is fiber rich, the mucus layer, which is in pink, is very high. If you have a diet which is fiber free, the mucus layer in pink is very, very narrow. Now, if you have a pathogen which is arriving in the intestine, and these are the yellow bacteria which are on the left, they arrive in the intestine, and you see that the mucus layer will really prevent the pathogen to go through the mucus layer and to go to the epithelial cells. If you have no mucus layer, the pathogen will be able to go through. So this is an example where the diet is really inducing a protection against pathogen. So the composition also decreases with age. And the composition decreases with age, and also the richness of your microbiota decreases with age. Finally, obviously, the microbiota is affected by antibiotics, which kill bacteria, But what people don't know is that the microbiota is also very affected by non-antibiotic drugs. If you take, for example, antidepressing drugs, they are also affecting your microbiota. And the microbiota is really playing a role, in fact, in the metabolism of the drugs that you are taking. If you are taking some drugs, they may be stocked in your bacteria, or they might be somehow catabolized by your bacteria, and they may have different effects in different type of population. So what is clearly shown now is that the microbiota is really affecting the immunotherapy in the case of cancer patients, and it's more and more clear that the microbiome composition is really affecting both the immune response and both the immunotherapy effectiveness in patients. So this is true for the cancer for the response to cancer. But what I would like to say today also is that the microbiome is also affecting autoimmune diseases. So this is a slide is presenting you a very, very famous slide, which has been done by my friend Jean-François Bach, who died a few days ago, where you see on the left that there is a dramatic decrease of infectious disease and a dramatic increase in autoimmune disease in the recent years. And for that, the hygiene hypothesis proposes that the lack of early childhood exposure to infectious agents, gut flora, and parasites increases the susceptibility to autoimmune and allergic disease by modulating the immune system development. So clearly, children which have been in contact with infection agents, gut flora, parasites, are more immune to other infectious disease, and they are less susceptible to autoimmune disease. And this is why my friend Brett Fidley proposed this book, Let Them Eat Dirt, just to favor the development of the immune system. Sorry. So in this review from 2022, the authors have really shown the impact of the gut microbiome on extraintestinal autoimmune disease. What they have really shown is that patients with extraintestinal autoimmune disease such as MS, rheumatoid arthritis, type 1 diabetes, and systemic lupus erythematosus have distinct gut microbiota composition compared to healthy control. More than that, they have shown that fecal microbiota transfer in animal models for autoimmune disease really reinforce the hypothesis that the gut microbiota is playing a key role either in the immune response or in the fate of the autoimmune response. So what to do? So you can somehow modify the microbiome by various approaches, which are shown here. Either you can add strains by giving probiotics, or you can eliminate some strains in the microbiota by giving some antibiotics, or you can alter the abundance of existing commensals, either by the diet or by prebiotic, or you can directly replace the microbiota by fecal microbial transfer. So I think I am not going to go into details, but clearly the gut microbiome is really playing a role in the autoimmune disease. And I think my take-home message is the following. There is a growing body of evidence which suggests that the gut microbiome may be a key factor in influencing predisposition to autoimmunity and allergic disease. So the challenge for the future are what? They are the understanding of how some bacterial species of the gut microbiome affect immunity, and the search for those very specific bacterial species is really the goal of many laboratories in the world. And second, the identification of the factors which will affect most the composition of the gut microbiome. So the gut microbiota might be modulated in patients with autoimmune disease as a potential preventive or therapeutic approaches. Thank you.
Thank you so much, Professor Pascal Coussat, for your insight in microbiology and infection biology. So, with her presentation, we're coming through 4 presentations. And now, the 5th one is the closing with the first part presentation of our symposium today. And now let us welcome to the stage Assistant Professor Dinh Van Nguyen, MD, PhD, Head of the General Medicine Department of Vinmec International Hospital. And with over 10 years of expertise in allergy and clinical immunology, he specialized in asthma, allergy, and related diseases. And his presentation will be in the diagnosing and treating autoimmune diseases in Vietnam. And Professor, stage is yours.
Thank you so much. And good afternoon, distinguished professors, scientists, and colleagues. And I'm very happy to be here to present about the unmet need in clinical practice in autoimmune disorders in Vietnam. But I think it's very hard to follow very excellent presentation from our distinguished professor. So before starting, I would like to say thanks for VinFuture, Vingroup, and give me a chance to be here. And let's start with the first slide. And you know about the autoimmune diseases, that the condition in which our immune system mistakenly attack our healthy cells. That means can make diseases and manifestation, clinical manifestation. And autoimmune disorders can be divided into 2 types, the systemic or organ-specific autoimmune diseases. And you can see here on the slide, and so mainly that the thyroiditis, that like Hashimoto thyroiditis and Graves. This is very common. And another thing is psoriasis, that in dermatology. And the third one is the type 1 diabetes. So it's very common. So for systemic autoimmune, you can see here, the mainly the Sjögren and lupus erythematosus, scleroderma, and vasculitis. So regarding to prevalence, and previously, they were considered autoimmune disorders by rare, but now it's not. It's not rare. This accounts for 3 to 5% of general population, especially after COVID-19 pandemic, and we got an increasing rate of the autoimmune disorders. And you can see in clinical practice, but we can have evidence from the research from UK, US, and other countries as well. And the relative risk increased by almost 50%, the relative risk. So very, very high. So that's a big problem for our healthcare system. And in Vietnam, unfortunately, even though we have a very big scale of the population, almost 100 million, but we don't have data on the prevalence of autoimmune disorder. But based on other study outside of Vietnam, they estimate that we have a 4%. So we have almost 4 million patients suffer from autoimmune disorders. So that the question is that how we can do for Vietnamese population. So now time we can do like an epidemiology research, and we can provide information for clinician, for policymaker, and for scientists, and we can know how to do for the autoimmune disorder in Vietnam. And here, back to the model of the pathogenesis for autoimmune disorders, and taking lupus erythematosus, for example, and back to the Professor Chi presentation, we know already about how from the healthy person to become an autoimmune patient. And from Professor Crossan, we know already about the environmental triggers, that are microbiota, epigenetics, but based on the patient with the genetic predisposition. So that's a very high risk and trigger our immune system to create autoimmune antibodies. That's the first phase. And the first stage of the autoimmune autoantibodies and cytokine production. And afterward, we can get the pathogenic autoimmunity. Pathogenic means there's some clinical manifestation, but it's incompletely presentation for the autoimmune completely with the criteria. So by the time autoantibody and production of cytokine increase and make the clinical manifestation, and we can divide the patient in different kinds of diseases like lupus erythematosus or scleroderma by different manifestation, clinical manifestation, and endotype. And look at this, and we can see here we have a different time. We have a time to prevent, and as Professor Grossen said, we can invent and we can change our microbiota on skin, on gut, and on airways, for example. So we can change our immune response, and we can stable our immune system, and it's to prevent for the patient to become an autoimmune patient. And we can have the first step, prevention, the second, and the third prevention, that means we can prevent for the more damage in our organ. So we can treat the patient. And today we can focus on treatment as well, and later on I can explain about how we can do that. And here, taking lupus, for example, we have a lot of things here for the challenge. And we have a challenge in terms of treatment. We have a challenge in terms of finding the biomarker to define the patient at the early risk, timely treatment, and following up the treatment for the patient. We also have a challenge in terms of managing our patient in pregnancy, in pregnancy period. And we have also the high comorbidity in the patient with SLE. And we also have medication. And later on, I will explain about even though over the world we have a problem with the medication, we cannot curable patient with the systemic lupus. However, in Vietnam, we are lacking of a lot of standard medication and biologic therapy. Here, to be focused on how we can define the patient at the early risk for the classification and prevention. And look at this. Now, currently, we are working on this autoantibody that is our current biomarker for the autoimmune disorders. Even though we define a lot of autoimmune antibodies here, but almost only play a role at in diagnosis of the patient, prognosis of the patient, and define the clinical phenotype and endotype. But it's very, very rare. Autoantibody can be used as a predictor. So it's very important for us to define and to find new biomarker. And look at this, and here we have from our population with the heterogeneous cluster. But we have to define for homogeneous cluster later on, and we can justify the risk, and we can prevent earlier and timely treatment for patients. That's very important now, and the question for all Christian scientists and policymakers as well, how we can do that. And here, another thing in clinical practice, with the autoimmune disorder that is cross a lot of specialty. So we need to MDT team to work together and to find out the best solution for patient with autoimmune disorder. Exactly like MDT team for the patient with cancer. And we have to take care of all the aspects of patient. Because, because the fatigue, 1/3 of patient having the fatigue and anxiety as well. And so we need to treat the patient not only for medication, but also for the healthy and mental health as well. And we have here the 25 patients having more than one autoimmune disease and multiple autoimmune syndrome that more than 3 diagnoses on patient. So that's a very complicated and very high mortality in the patient with autoimmune disorders. And finally, I would like to mention about the treatment. And here we can see the history of treatment. And from ancient time, we have only based on herbal and plant products, like a colchicine and like a mycophenolic acid, and afterwards, the aspirin and quinine and for patients with inflammation. But now, we based on the corticosteroid and DMARD, that's the disease-modifying anti-rheumatic disease medication. So it's very helpful for DMARD to sparing, as a sparing agent for corticosteroid and to reduce the risk for the adverse reaction for the patient with the corticosteroid. But we need biologic, we need new therapy with the targeted therapy. And look at this slide, and it's very complicated, but I would like to show, because here's in IS, that means immunologic synapse, and that's working together with B cell and T cell. And Professor Chi mentioned already, and Professor Sakaguchi mentioned about the T rate and how to work in inhibit B cell and T cell autoreactive. So we have a lot of approach here, like we have a molecule on the surface of cell, we have a contact interaction between cell to cell, and we have other cell here can be inhibited. So a lot of options for us for the further treatment and for the new therapy, that's targeted therapy, or the biologic therapy. But some of them has been approved by FDA for autoimmune disorder diseases, autoimmune diseases, or lupus erythematosus, but unfortunately it's not available in Vietnam. So now I hope that policymakers can make that happen and available in Vietnam. However, it's very difficult to approach because it's very expensive. And in the US, then one-fourth, 25% of patients having problem to pay for the medication. But in Vietnam, I guess that's a very high prevalence as well. And we estimate only less than 1 to 5% of the patients in Vietnam can be treated by biologic therapy. So I think we need to find the option and find the solution for Vietnamese patients can be affordable for the new therapy and to get them better and curable for their diseases. Thank you so much.
Thank you so much, Assistant Professor Dinh Văn Nguyen, for your enlightening presentation about the unmet needs of diagnosing and treating autoimmune diseases in Vietnam. So we've come through the first part of presentation with 5 speeches. And now we are coming to the second part of today, panel discussion. And now it's time for us to welcome back to the stage Professor Chi Van Dang to come to stage to moderate the discussion. And now I would like to have the presence of distinguished guests. They are also the panelists today. Please welcome back to the stage Professor Jiang Su-chun, Professor Pascal Cossart, Professor Shimon Sakaguchi, and back again on the stage, Assistant Professor Dinh Văn Nguyen. Please welcome back on the stage all our professors. Thank you so much. And you are coming to the second part of today's symposium, panel discussion, where we could share the experiences and the thoughts, the opinions, and discuss more about the topic today. And now it's time for me to give the floor to Professor Chi Van Dang. You will moderate the discussion. It's your time. Thank you.
All right, so we're going to be soliciting questions from the audience. So if you have questions, please come to the microphone and ask your questions. But maybe I can just start with a general question for all the panel members, ending up with the last talk, speaking about technologies that are affordable in developing countries in general beyond Vietnam. So if you can reflect on your area of interest, maybe I can start with the Tregs, say CAR Tregs, which as you know, with CAR T-cell therapy for cancer, it's still not very affordable. Where do you see that going in the future, and maybe perhaps reflect a little bit on whether there are any roles for bispecific antibodies at all?
Well, so CAR T cells, so it has been shown that it is effective in certain cancers, especially B-cell lymphomas, B-cell leukemias. And then now people are extending that success to other cancers, especially solid cancers. But now, but not so successful yet. The reasons for that is, so CAR T, so it has T cells engineered to the one specificity. So recognize one antigen, for example, B cell expressed antigen, so all the B cell lymphomas or B cells all wiped out. So that kind of, it's difficult to find out every cancer to find out that kind of the target molecule. So that is one problem. And second one is when these T cells, when they go to the cancer's tissue, they become it is called exhausted. So they lose function. How we can overcome this, that is another problem. And third one, just recently reported, that is we manipulate the genome, DNA of T cells. And then sometimes you might see lymphomas. So that is a kind of adverse effect. So these findings, well, so if you compare with the previous approach, for example, the bispecific antibodies or other monoclonal antibodies, which one is better or more effective? It is still controversial. And each technology is being developed, improved. But we'll see what is which one is better, or not only scientifically or medically, but also economically. CAR-Treg from individual cancer patients, it costs a lot. How we can reduce the cost, that is another problem. So, and if that technology is applied to regulatory T cells, so regulatory T cells become engineered to suppress particular immune response, for example, autoimmune disease, perhaps we may have a similar problems.
Well, thank you. Maybe I can turn to Professor Kozart in just reflecting on the same type of question on what do you think it would take in terms of research that allows us to modulate the microbiome, particularly for autoimmune diseases, whether it's dietary manipulation or pharmacological manipulation, aside from fecal material transplantation. Any thoughts on what it would take for us to get there?
Maybe I don't answer directly to your question, but I think the key issue now is to identify what are the bacterial species which are responsible for a good response in this organism or for a bad response in another organism. I think we are still at the infancy of a field where it would be nice that all groups agree that this very bacterium is responsible for a good response in all patients, or why it is not universal in all patients. I think this is the key issue. So now, let's say we identify bacterium X as a good bacterium for the immunotherapy. Well, either you re-inoculate this bacterium to a patient if he doesn't have enough of this bacterium, or you try to give a diet which would favor the increase in the concentration of this bacterium. But really, we are still in the infancy of this field for the moment.
So what do you think about the geographic differences in the microbiota, right? Different parts of the world. What would it take, for example, you already mentioned that different areas might have different microbiota. Yeah. How do we deal with that complexity in dealing with therapy from that kind of knowledge that's going to be specific to geography?
Yeah, okay. So I think it's a good point that the composition of the microbiota is very, very different in all countries and all that. And really, we are very close from the diet problem. So I think we would have to deal with that. Yeah, I think it has to do with the diet, because we know that the diet is really responsible. There has been studies, for example, which have been done with twins. you know. And if the twins were really in different countries, then the microbiota is very different. So we can eliminate, for example, the genetic component.
So maybe I can turn to Professor Chung. And you mentioned about the use of, say, antibodies against cytokines for control of certain autoimmune diseases, which has been successful. And then I have two questions. One is, what are the current limitations that could there be further development of those type of drugs? And then the second question is, you mentioned a little bit about how to manipulate transcription factors such as HIF2 alpha, for example. So for the first question, are there any questions remaining for developing new antibody therapeutics that you can think of for development? Because in general, antibodies are more affordable than cell engineering.
Yeah, I think most of the biologic DMARDs are developed against the cytokines, and almost all cytokines are already tested. So, and actually not all biologic DMARDs have effects on autoimmune diseases. So we may need more biologic DMARDs against many different kinds of cytokines or chemokines. So we have to expand the target. And we also have, as far as I know, we have antibodies against the various immune cells to the specific antigens. So I think we need more targets on the specific antigens to develop more monoclonal antibodies or recombinant proteins.
So in terms of developing, say, HIF2 alpha inhibitors that you mentioned about, what are the directions that's going to need to be taken for that? Because some inhibitors have now been approved for renal cell carcinoma. So clearly it's been used in humans. What do you think it will be? What would it take to actually implement that in a rheumatologic disorder?
You know, HIF2 alpha is nucleus proteins, and G8 is membrane proteins. So the development of biologic DMARDs, I mean the monoclonal antibodies, may be very difficult, especially for their delivery into cartilage tissues or joint tissues. So we have to, I think, we have to focus on the development of targeted synthetic drugs if we are going to develop targeted therapies against HIF2 alpha or G8.
Then I'm going to turn back to Professor Din. You know, you mentioned about sort of challenges that are in Vietnam right now. The study of the prevalence of different types of autoimmune disease needs to be identified. But beyond that, what do you see are the current therapies that are available clinically beyond say, high-dose steroids or low-dose methotrexate for rheumatoid arthritis? What are some limitations to use some of the antibodies that are therapeutic antibodies that we just discussed in Vietnam?
Yes, and I think currently we are being used like a medication generally in Vietnam, like a corticosteroid and DBAT and immunosuppression agents. And so, you know, already we know about that very high adverse reactions and the high risk for the inflammation by 10 to 15%. So for biologic therapy, we are also using for some already approved by government for patients with like anti-IL-6 or like a CD20 molecular antibodies, and but it's still very expensive for Vietnamese patient. So I do hope we can do like we can have like a company. They can use a local facility, and we can produce the medication here in Vietnam that can lower price, and more patients in Vietnam can be affordable. And I think that's another way. And yes, you mentioned about the Gera factor for microbiome and other things, and genetic signatures from different kinds of ethics, right? So I think we need to like run a study in Vietnamese population, and we can find out the signatures and find out how different from the other population, and we can have a target therapy and exactly tailoring for the Vietnamese patients. I think so.
Great, thanks. We can open up the session to questions from the audience. We have. We need a microphone. We have a microphone to the front.
And now, yes, it's time for the Q&A session, and I will assist you to address the questions from the audience. The audience, please keep in mind that you have 1 minute to ask the question, and the panelists will answer in 3 minutes. Okay, now let's. Yes, Professor.
Yes. So I have a question. I'm trying to understand if you can quantify the two aspects for autoimmune diseases. One is what percentage of the autoimmune diseases are due to the microbiome defects, and what could be attributed to epigenetic effects? Do you know those?
So, Professor Chivandang, who would you address this question for?
It seems like the question is directed to you.
Well, it's addressed to me, but I don't have the answer. I mean, it's clear that we are on our way to demonstrate that the microbiota is playing a key role. But clearly, to say what is the role of the epigenetic compared to the microbiota, this is impossible to say.
Thanks.
Yes, please. So, staff, could you assist me a mic for the first row? Yes. The gentleman right here at the first row. Thank you.
So I would like to thank all speakers for your very interesting presentation. My question for you is concerning the CAR T cell for autoimmune disease. As we make now, we can produce CAR T19 and treating lymphoma and acute lymphoblastic leukemia successfully. And as you know, recently one Parkinson team in Germany, they perform a clinical trial phase 1 using CAR T-cell 19, CD19 for lupus. That's why we plan to perform clinical trial phase 1 using CD19 CAR T for lupus in epidemic. Here now we can produce CAR T-cell with the cost and also including other treatment. The whole cost around $80,000 It's much less than to compare to cost in US or in Europe. Before starting, we would like to ask you about your opinion about the efficacy of CD19 CAR T for lupus. Thank you very much.
Yes, I think if I understand the question correctly, is that you have the capability of making CAR-19 and that you can direct it for autoimmune diseases to eliminate B cells, right? Particularly for autoantibody-driven autoimmune diseases. If I understand it right, in terms of how do you further develop that or educating about that broadly, maybe that's question for you, Dr. Din, and how do you disseminate that in Vietnam? I still have a question about the economics of that because there are some institutions that try to make it at cost if you have the right cell production facility to really produce it in an affordable way, but perhaps maybe the question is directed.
Yeah, I think I can answer because the Professor Liam now running CAR T19 for the patient with cancer. It's a patient with hematology cancer, but now he would like to move to treat for autoimmune disorder like lupus, and we based on the evidence from the Germany hospital institution. They did already some cases, but I think. To answer for that question, I think Professor Sakaguchi is the best professor can answer because you are running a project on CAR T rate and T rates in autoimmune. So I think.
Well, so CD19 CAR T. So it is very effective in the killing all B cells and then also B cell leukemias. But as you imagine, the side effect is immune deficiency. So the antibody-dependent autoimmune diseases such as lupus, and then depleting all B cells for long term, you can induce remission, of course. But can you deplete B cells for the rest of life? That is the question. So theoretically, of course, without B cells, and if you put the patient into the clean room, and then patient can live in clean room the rest of his or her life, that's possible. And then, of course, they can survive. But practically, it's not a good idea. So I do believe that the depleting B cells by CD19 CAR-T for inducing remission, that's okay. But then you must replace with other treatment. Otherwise, it's not treatment. Right.
Thank you so much, Professor Shimon. Yes, and that is the last questions and the last answer for this symposium today in the second session. And don't forget that you are provided with the hand with the paper so you can write down the questions and all your hesitation that you want to send to the panelists today, and we will collect and send it to the panelists and we will answer in the paper. Thank you so much for your joining. And once again, thank you so much for Professor Chi Van Dang and all the panelists today. One more time, please give a round of applause for them. Thank you so much. And ladies and gentlemen, thank you for your attendance and your support during the Win Future Prize 2023. And don't forget that on the 20th December 2023, the main ceremony of Win Future Prize will be live streamed on VTV1 at 8 p.m. and we will find out who the winners of one of the most valuable prizes in the world, and we look forward to your presence. Thank you and goodbye for now. Thank you so much.